In this lecture, we focus on some key beer flavor molecules and the yeast metabolic pathways that lead to those endpoints. And I'll try to emphasize how these are affected by the grain bill, yeast strain, and brewing conditions. I'll mostly use the term flavor here, and for simplicity's sake, I will rarely make the distinction between aroma and taste. In the end, I'll return to our expanded set of brewing microbes to briefly discuss wild fermentation and the microbial progressions that occur in uncontrolled brewing conditions. Here's this list again, this time with a limited set of flavor outcomes presented. From this really simple list, we see a substantial slice of the flavor possibilities in beer: esters, phenols, organic acids, ethanol, sulfite, as well as fermentation conditions such as temperature. And we get the general sense that different fermentation and flavor outcomes can be controlled by the yeast species or yeast strains, as well as other microbes. This is just a rogues gallery of flavor types and examples, and is by no means comprehensive. Most of these would be considered off flavors, except when they're not. I think rancid butter, however, is an obvious no-no. However, hydrogen sulfide, and more importantly, dimethyl sulfide, or DMS, is worth mentioning. The sulfur smells in beer mostly derive from the degradation of sulfur-bearing amino acids in the grain, principally methionine. Both these compounds are quite volatile and are easily blown off during the boil stage of mash-in. DMS does appear in limited quantities in some styles, though, particularly lagers. And recall that one of the qualities that sets Saccharomyces pastorianus apart from ale yeast is its enhanced sulfur uptake. No surprise then that these beers might retain some sulfur component in their flavor profiles. Recall too that sulfites are a flavor stabilizer, as they inhibit the establishment of microbial growth that could alter the collection of flavor molecules. It's interesting to consider whether a style flavor parameter, mild sulfites in lagers, might actually have been driven by the necessity of using a strain that naturally slowed spoilage. Moving on, diacetyl is almost always considered a negative, except for some British beers, particularly BJCP categories 13 and 14. Remember that BJCP stands for Brewing Judge Certification Program, and that's our standard list of beer styles that we refer to in this course. Isoamyl acetate is also a negative, save in Weissbiers, or wheat beers, especially category 10. Later in this lecture, I'd like you to recall that I mentioned that the isoamyl group is a branched amino acid metabolite. It may be that wheat in the grain bill can affect protein availability and thus amino acid availability. More of the amino acid precursor usually means more of the fusel alcohol and its esterification products in the beer. Likewise, phenolic flavors are usually not desirable, but again, they appear and are even desirable in whites and wheat beers, as there's wheat in the grain bill. In this case, though, we'll see that it's not increased protein, but the presence in wheat of the woody cell wall polymer, lignin, that gives rise to increased phenols. The particular molecule shown here is 4-vinyl guaiacol, which has a clove aroma. Swap the vinyl group for an acetaldehyde moiety, and you got vanillin, the phenol principally responsible for vanilla's aroma. Generally, the fusel alcohols are negative flavor components and add a solvent-like or hot note. Keep in mind, though, that these major categories of flavor molecules hide a great diversity of individual and idiosyncratic structures. Okay, big picture. Inputs are blue, sugar, and amino acids. Glucose feeds the pyruvate and keto acid pool, and amino acids feed the keto acid pool too. All those same major outcomes are presented here. Ethanol we're already familiar with as a product of pyruvate. Acetyl-CoA leads to fatty acid synthesis, which lead us to esters, a critical flavor endpoint, as we'll see. Acetyl-CoA also leads to the keto acid pool. We'll see in a moment how amino acids and acetyl-CoA can drive formation of fusel alcohols and thus to esters. Phenols are also shown here, and it's clear that they're derived primarily from the medium, as I mentioned, from the lignin present in brewing grains, a major phenol contributor. Finally, let's stop at diacetyl, which is a pyruvate decarboxylation product. This is another off flavor that, like sulfur, can usually be easily avoided. Diacetyl is produced by the yeast and leaves the cell during primary fermentation in the attenuative phase. However, it can be beneficial to leave the beer on the yeast trub for a couple of days. This is because as the cells enter stationary phase, they reabsorb the diacetyl, reducing it to acetoin and 2,3-butanediol, which, with flavor thresholds between 10 and 40 parts per million, are much higher flavor thresholds than diacetyl's 100 parts per billion. Stepping back out, the main energy production and primary metabolite pathways lead more or less directly to most of our flavor outcomes. Looking closer, many amino acid pathways lead to higher aldehydes, which are themselves strong flavor compounds, and thence to fusel alcohols. Fusel is a German term of unknown etymology, meaning bad alcohol. The key route from amino acid to fusel alcohols is called the Ehrlich pathway. German scientist Paul Ehrlich was isolating amino acids at the beginning of the 20th century, and he noticed structural similarities between isoleucine and isoamyl alcohol. In nutritional supplementation studies with yeast, he discovered that the presence of the amino acid does indeed drive production of the corresponding alcohol in yeast. Valine, isoleucine, methionine, and phenylalanine all form alcohols along this pathway. Oh yeah, and let's not forget that he also won a Nobel Prize in 1908 for his work on immunology, was a pioneer in cell staining methods, and basically invented chemotherapy. He screened small molecules for activity against a variety of pathogens. His colleague, Hata Sahachiro, identified the first such agent for syphilis in Ehrlich's lab. That drug, salvarsan, remained a useful treatment for decades until the advent of penicillin. Anyway, in the Ehrlich pathway, amino acids are deaminated to the corresponding keto acid, and from there decarboxylated to the fusel aldehyde. Reduction by one of many alcohol dehydrogenases leads to the higher alcohol. Recall from previous lectures that ADH1, the ethanol production engine in yeast fermentation, has activity against a range of other higher alcohols. The takeaway is that amino acids and the keto pool lead more or less directly to higher alcohols. Returning to the origins of the term fusel, we know that these molecules affect the flavor profile of beer, but not in a good way, adding hot or solvent notes. What we'll see next is that these alcohols are not an endpoint, but lead to esters and ketones. In this slide, I've placed some key beer endpoints and their flavor thresholds in parts per million. But overall, I'd like you to notice that in the Ehrlich pathway, the aldehyde is oxidized to an alcohol with a concomitant reduction of NAD+ to NADH. This is distinct from the acetaldehyde to ethanol reduction, and an alternative fate of an Ehrlich aldehyde is their oxidation to a fusel acid, and that reaction regenerates NAD+. We are staying focused on the volatile and much more flavor-relevant fusel alcohols. In fact, there are many pathways leading to flavor molecules, of which Ehrlich is only one. This slide shows the most important routes. As mentioned, the Ehrlich pathway draws from both the TCA cycle and amino acid biosynthetic routes, leading to fusel alcohols. Production of acetate and from there to acetyl-CoA from acetaldehyde supports the formation of acetate esters of Ehrlich pathway alcohols, as we said, with ethanol itself producing ethyl acetate. But higher order alcohols support the formation of a diverse set of acetate esters as well. Condensation of two acetyl-CoA units leads to malonyl-CoA and from there to medium-chain fatty acids, which are subject to esterification with ethanol to produce flavor-critical ethyl esters. Post-fermentation, these are often considered to be off flavors and not desirable. Now, brewing conditions can affect the outcomes of the Ehrlich and associated ester pathways. Increased sugars has complex outcomes, but many are considered to drive acetyl-CoA production. Increased nitrogen availability in the wort leads to increased amino acid availability in the cell, as we've seen, and elevated brewing temperature drives expression of yeast membrane permeases that lead to increased amino acid uptake by the cell. In both cases, the Ehrlich pathway is fed, leading to more fusel alcohols and more esters. Often, however, the outcomes of these kinds of manipulations are unpredictable in terms of which esters are produced and can lead to unbalanced flavor profiles. This means overproduction of flavors that might otherwise be considered positive. So it's possible to prime yeast ester production at the beginning of the process by adjustment of the wort composition and at the end by adjustment of fermentation temperature. But it's important to recognize the challenges of these manipulations as well. Now, the rightmost category in this slide is actually a bit of a digression, since most of the beer-relevant molecules in this category are hop-derived. But since we're discussing flavor metabolism, it's worth mentioning the terpenes. First, let's be clear. The molecules have strong odors, as the name might imply. They were initially recognized as components of turpentine. Today, turpentine is a petroleum product, but historically, turpentine was a plant-derived substance. The name is of Greek origins, meaning resin, and its eponym is the terebinth tree. Anyway, terpenes are also a broad set of important flavor molecules generated across taxa. For our purposes, they're a distinct endpoint for acetyl-CoA, where condensation of 3 acetyl groups leads to mevalonate and eventually isopentenyl pyrophosphate, and from there to the isoprene unit itself, which is the building block for the monoterpenes, referred to as geranyl or isoprene times 2, the sesquiterpenes, referred to as farnesyl or isoprene times 3, and the diterpenes, referred to as geranyl or isoprene times 4. I'm actually not going to come back to these in this lecture, but the compounds will be of interest in the next lecture reviewing hops. What I can say about terpenes and yeast is that research groups are actively engineering yeast strains to produce some key hop terpenes by collecting yeast genes encoding highly active enzymes in the terpene biosynthetic pathway and combining them as a single strain.